Thanks, Karen. And maybe you could enter your email in chat so that if people want to sign up and learn about the trainings, that would be a great way to do that. All right. Well, now our next speaker, Elizabeth Boedecker from St. Louis is going to, and T. Wen from St. Louis as well, are going to talk about developing coursework in targeted genome editing.
I'm muted. Anybody who had that on their bingo card, bingo. My name is Elizabeth Boedecker. I'm the director of the Center for Plant and Life Sciences campus of St. Louis Community College. T is senior scientist CRO coordinator that is working with us over the last year. And so we received a grant around developing genome editing coursework towards the end of 2019. I don't know that we actually received the funding until 2020, but right in time for all the fun stuff we've had over the last 2.5 years, which has definitely impacted our ability to get this project moving. But basically, we had sources in industry and academic backgrounds that were coming to us, specifically a couple folks at the Danforth Center, that basically approached us a few years before we received the grant, saying that they're doing a lot of genome editing research, and it would be really awesome if there was some way that we could get some additional training for our workforce, for our students in our biotech program, a little bit of training around genome editing, because it could make them a much more valuable asset to the folks that are potentially hiring them. And so we started exploring, how to go about applying for some funding from ATE in order to basically help us develop this, because there was going to be some expenditures related to equipment and stuff that we thought we could definitely be utilizing from the government. And so this is from today, like an hour ago. And this is a trend that we've been seeing over the last 2 to 3 years, even throughout the course of the pandemic. Bioscience industry in this region is booming, and we've actually seen growth over the pandemic, and it's just exploding now, even worse than it was before. And just specifically related to genome editing, if you just search for genome editing on Indeed, there's 41 local jobs already listed. And if you look at other websites, there was one that I looked at that had 79 jobs in St. Louis that specifically mentioned genome editing. And so this is very much something that is needed. And some of the, this is not just 1 job or 5 jobs. This is, dozens of jobs that are listed right now today. And companies that are within our building, across the parking lot from us, these are just a few of the people that we work with that have jobs posted specifically citing genome editing as something that they're interested in hiring someone with some skills around that. And so this actually already fit really well with the biotech program that we offer. And so we already have a pretty robust curriculum. We have a couple of tracks where you can get the applied science degree, or if you already have a life science degree, you can just come do a certification in biotech with us. And so we already teach a lot of things about the DNA structure, the function, a lot of the things listed here, how to isolate DNA, how to maybe construct a plasmid, do some cloning, design some PCR primers, how to sequence stuff. We include all of that in some of our core curriculum for the program. And so, as we are all very much aware, bioscience, wow, it was moving fast before, and it just moves so quick. We, years ago, had implemented a way that we could actually be offering new technologies, new techniques, new things that were occurring in bioscience without having to go through the 3-year process of getting curriculum approval. And we basically teach these under our advanced topics heading of Bio 226. And we can offer different kinds of training under that advanced topic heading. So we actually have bioinformatics, we have forensics, proteomics, quantitative PCR technologies, plant transformation, RNA interference, genome editing is going to fall under this same Bio 226 heading. And this allows us to implement maybe some new advancements in bioscience without having to have a delay before we can teach it, because we have to get it through some curriculum process. So this allows us a lot of flexibility in what we're able to teach, what we're able to offer, and how quickly we can get it on the schedule and students being trained. And so once we received the funding, we did really want to go through a pretty rigorous process. And throughout the whole history of our biotech program, because our landscape here is ever changing, and it's very much a booming startup company area for us right now, we never want to just be solely focused on what one company is telling us. So we didn't want to just listen to what Danforth says they needed, because that's not how we've developed a successful program. The successful program comes from working with all of the industry partners in the area and having them identify what they think are important skills that our technicians should have coming out of our programs. And so we wanted to do the same thing with genome editing, because it's very, there's a lot. When you say genome editing, just kind of when you say bioscience, that could mean a lot of things. And so we really wanted to sit down with a cross section of our industry and find out what it is that they mean by that. What kind of skills in particular are they looking for a student to have if they've completed these genome editing courses with us? And so we wanted, like everything else in our program, we want this to be hands-on experiences for our students. We want there to be, ways that the industry can then save on their training time and costs if they hire one of our students. We want to take other industry-ready folks, like graduate students who maybe don't have all the same level of lab skills, and get them upskilled in genome editing also. And with the idea being that, if you are successful in genome editing, it really is kind of indicating that you have many different skill sets that you're kind of implementing into that field, because it encompasses so many different aspects. And so we definitely wanted to consider all that in creating the course, and we're actually planning on doing genome editing 1 and 2. While I'm not providing an excuse here, it's more of an explanation. We had things going. We had things rolling really great when we all went down into shutdown mode. And we have really struggled getting people involved coming back from that. And so we are definitely going to be looking at a no-cost extension, potentially even an additional year after that, because part of what was included in our grant funding was funding for internship to support in genome editing specifically, because we have enough industry partners locally that are doing genome editing. They want to provide internship support so that once we have some of this training under these students' belts, they can actually go out in the industry and practice it in a special workplace learning component. And so this is something that I don't anticipate we're going to get spent down over 1 year's time. So I am expecting an additional extension beyond that if it's possible. And I have to talk to my program officer about that. But this way we are able to, the way we've done this, we're able to really update that curriculum as we need to, as there's changes in the industry, and always be offering what is happening in the field right now, because that's really the struggle, right, is to stay current in what is happening. And so we want to make sure that what we're teaching is producing folks that are well qualified for what we're asking them to do and claiming that they can do. And so the things that we are including in our Genome Editing 1, our advanced topic, which is going to be an 8-week, 2-night-a-week class, we're talking about the history, because our industry has stated to us, people need to understand how we got where we are today. And so part of that is training them on how these developments occurred and the history around all these genome edits related to health and plants. What are the different types of tools available? How do you go through designing your editing process? How do you detect the edits once you've made them? what are the bioinformatics components surrounding some of those other techniques that I just mentioned? And then a really big piece, and I guess this shouldn't surprise me, but I think it did a little bit, is that there was a lot of concern that they wanted to make sure the ethics of doing genome edits was very thoroughly examined over the courses that we're going to be teaching in this topic. And so the ethics and regulatory processes are heavily ingrained in this whole curriculum that we're designing. And then we're going to be, talking to actual researchers, talking about career options within this genome editing field, talking about case studies. And then as everything else that we do here, there's going to be labs that are involved. And so that takes an enormous amount of planning, testing the rigors of those labs, thinking ahead about how to plan for them so that things are in place that when the students are ready to do, some guide RNA design, that, we're ready to do that. If we have agro ready to do infiltration with plants, when the gene gun is going to be available. And this is one of the big things that, one of the big pieces that we got pretty specific feedback from that panel that we pulled together to ask about what we should be teaching, where initially we had placed purchasing a gene gun in the grant funding. But it became abundantly clear to us that the industry didn't think that that was really necessary, that we could go get, a minute on someone else's gene gun, because they really thought there was a lot of other ways that were way more high throughput than a gene gun would be. So that was definitely an adjustment to our funding and what we decided to use it for. Other things like protoplasts and of course worms. I love my C. elegans, so I want to use worms for everything. But so there's all kinds of things that are being kind of laid out in this curriculum. And so I'm actually think I'm going to pass this off to T, even though my picture is on here. I think I'm going to pass this off to T because she's really been the one that's worked with our industry partners, Marianne and Susanna, to really design this. So I'm going to let T take it from here and talk about the actual lesson plans.
All right. So as Betsy was saying, one of the things that we're trying to do is because the way the course is structured, where we're meeting twice a week for 4 to 5 hours a week, we wanted to see that we could do the experiments in that timeline. And so in working with our industry experts and researchers and looking at the skills that have been requested by industry employers, we set these lesson plans together. This is just the top part of an example, but we set the learning objectives and the skills and say for this one, it's 4 hours and we're doing an agro infiltration. How does that work? So can we test it? And what I think is really fun is that we currently are hosting high school interns. And so we're able to test out the protocols with the high school interns and see how that works. So next slide, please, Betsy. And so this is how we were planning it for the evening. And these are our course instructors over here on the right, Dr. Susanna Koshikova and then Dr. Marianne Kramer. And this is from a visit. We went to the Missouri Soybean Association because we're trying to look at all the different kinds of places that genome editing is being done that is relevant to plant biology. And we met one of the team there, and they do marketing and policy, intellectual property licensing and research. We got to see the labs, the fields, the equipment. It was really incredible. So all of this can go back into, we've captured some videos that we plan to post and some audio for some podcasts. So looking at sort of a comprehensive curriculum as well. But in this agenda here, what we're seeing is that it's around dinner time so we want to give them a chance to eat. So we are doing a lecture during that time and using lots of video demonstration as well. And then breaking up the experiments. And this is the plan we had. And then what we'll do is, if you can go to the next slide, we went ahead and we tested this with the high school interns. And so we have a tobacco plant here. And we ordered these a little bit older than I accidentally ordered these a little bit older than they needed to be, so they're a little bit tall, but it was a fun experiment. And the other thing is the greenhouse accidentally planted the wrong kind. These are already naturally GFP glowing plants, and so we had to use a plasmid where we could visualize the agrobacterium infiltration. So we used a ruby plasmid. And I'll talk a little bit about that too. But if you look at the picture here on the right, that is the greenhouses from the Danforth Plant Science Center. It's literally just a few minute walk from the building that we're in. So we're really centrally located for this kind of work. It's pretty cool. And then the large building behind that is the Danforth Plant Science Center. And so we walked over to the greenhouse and we're able to do this experiment. And what's, I think, meaningful about it is that they prepared the agrobacterium. They did the UV spec to look at the concentrations. There was a protocol that we were testing out. We were curious about how much time it would take, how much reagents we needed, how do we prepare ahead of time, right? And what did the students pick up? What were they excited about? What did they still have questions about? So we were able to do the infiltration one day and then look at the visualization over the next week. We looked 2 days later, 3 days later, a week later. And I think what was fun about it is, one, they just loved being in the space. They just loved being able to look at plants. They didn't realize that you could do genetic experiments with plants at this point. And so just even that exposure was really exciting. You know, the next time we were like, oh, do you want to look at the plants in the lab? They're like, no, we want to go to the greenhouse. Next slide, please. And one of the goals and challenges that were mentioned in that landscape analysis were, would students be able to, we want to teach critical thinking skills. And so one of the things we did is we showed them, well, okay, these plants, this is what it would look like if the infiltration worked and it glowed green, right? The left picture. And then on the right is the agro infiltration. We're talking about controls. So we did a water only control and we did one with agro. And then we did infiltration with a needle stick versus not to see how the spread worked. And we talked about the viability of the leaves afterwards. And one of the instructors, you know, she's like, we'll point out why they might die. And I was asking, was like, what are some of the reasons they might die? And it could be even as simple as, you know, they broke the stem when they were working with it, or, you know, you damaged the plant. So it may not even be from the manipulation that you're using, it's just the handling of it. And all of that is important in experimentation. And so I think that kind of hands-on work and learning is really critical. And the note about the ruby, being able to visualize the infiltration without fluorescence. So I was working originally with Dr. Koshikova, and she said, oh, I have a colleague who can, you know, help us with the ruby plasmid. And then so that's where I met Dr. Kramer. And, you know, she was like, let me know how I can help. And I said, actually, do you want to be a co-instructor? And so I think in developing relationships with industry, what I found is that many industry employees and collaborators are really eager to participate and learn and instruct because they missed that part of, you know, maybe their schooling, that they got to do a little bit of teaching. So it's been a real pleasure to work with different industry collaborators, and I'll talk a little bit more about that in the next couple of slides. Next slide. So then we also, so that one was a tobacco plant, and so then we also tried it in cotton plants. So it's very, you know, Midwest Southern. We've got the tobacco and cotton, and here on the right, Taylor Harris is a PhD candidate. And she was teaching them, so they got to practice the protocol, and now they're applying it and scaffolding the learning with a different plant. And here they've got pathogen-resistant tobacco and then control regular tobacco, and they're looking at different pathogens. So then they're able to then take the skill they learned, apply it, because a lot of the industry partners were telling us it's the skill of pipetting accurately, being able to measure and handle things, right? So we wanted them to get a chance to handle the materials and practice the experiment, but use the critical thinking of like, okay, if this was the setting and this was the question or the hypothesis, what do you think you might observe? And the controls for there. And then we also get a chance to talk about global impact or regional impact in different spaces that these different plants are grown. So that's another thing. Next slide. A large teaching philosophy of our group that we've assembled to lead this course development is around real world connection. and connecting it to the ecosystem of science. And so Dr. Kushiktsova, suggested, let's look at these readings, because in the early chapters, they go through the central dogma, and then it goes through a little bit of history of genome engineering, and then it starts to talk about the regulatory, the ethical, intellectual property aspects of it. And so this gives us a chance to really explore that as well. And we also are able to integrate that with our career conversations. The students are really interested. This is something that they actually said after they did the analysis, after they did the plant experiments. We had them read a section, first part of Codebreaker, which is about eight chapters or so, and then we talked about the history of genome editing, and they said we're changing the basic building blocks of life, because that was one thing that really initially interested Dr. Dwadna. And so then we had them, yes, it's the actual building blocks. So then we pulled out the mold, I forgot what these kits are called, and then we had them build the different amino acids and have conversations as they were building it. So really seeing how they're putting it together and then supporting that learning. Next slide. This is our list of industry collaborators so far. On the left column is Dr. Kushiktsova and Carlos. I forget his last name, but they have been really incredible in helping to develop the course. They will allow us to use their gene gun upstairs and do the demonstration. Down below are our partners from the Danforth Plant Science Center, Dr. Kramer, who's the other co-instructor, and then Taylor Harris, and then Dr. Del Percio. So they'll be doing some speaking. In the middle row, we have guest speakers from industry. And you can see they have backgrounds in biology, PhDs in biology, but they work in different industry at this point. And on the right, those are some of the scientists and stakeholders we've interviewed for our podcast, and then to talk about what industry is looking for and how to get a job in that part of the biotech sector. And then, right, so we plan to share as a course in a box. We have a TikTok channel going, so you can check out some of that activity there. We actually have one demonstration. For sake of time, I had pulled it up, but we have one of a scientist doing an agro infiltration. So in a minute, so you can see that put together. And then we have a podcast channel with some biomanufacturing interviews already from our other course. And we're going to add on some genome editing so that you can have some experience with the experts that we've gotten to talk to. You get to hear those conversations. And so we're happy to share any course materials and have continued conversations. And big thanks to our co-instructors who are also on the call today too.
Well, thanks, T and Betsy. That's wonderful. Interesting stuff. I mean, I worked on agrobacterium as a graduate student, and all the things you're doing, it makes, we just use toothpicks, scratch calico leaves, or sunflowers. So this is sounding just way, way beyond what we ever did. This is great. So I think we have a small enough group here. Before we get started on questions, I want to remind everybody that we do have an evaluation that we would love for you to fill out. If you have to leave, please do that before you go. And Betsy has also put her email in chat so you can get a hold of her. And why don't people, you can introduce yourself and ask a question. No questions? Well, I'll start. Oh, I guess we have maybe one.
A really basic question. What is the 8 in innovate bio? Does it stand for something?
Well, the word innovate spelled with an 8, but the reason it's capitalized is it's because it's funded by the National Science Foundation's Advanced Technological Education Program. And so we wanted to emphasize the ATE part of it.
What is it?
It's a grant funding program. Other questions? Well, I have a quick question for Karen and James, and I'm not sure who would be best to answer this, but I was wondering about with your second grant, what you're doing to reach out to industry.
Like in general, what's our strategy?
Mhm.
So we're really partnering with California Life Science because we have a good connection with Laura Lindbergh, who I think has probably been on some of these before, who is works with a large group of industry in the state. I think just very similar to what Betsy and T were talking about, are in a situation where there's lots of different companies. And so we're not interested really in focusing on one company. It's very rare that our students we really don't, we might have 2 students here and there's so many startups. And so it's really important to get input from a vast, like large companies, mid-tier companies, small startups. And so we've kind of gone through, like going through a trade organization is a more useful way for us. And so the next stage of the next time we do the trainings in May, we're hoping for somewhere in the realm of 30 to 50 participants from industry that then we can gain feedback from to move forward.
I have one other question, and then I'll answer some that I see in the chat, or ask some that I see in the chat. Karen and James, what do you see? Are you able to measure the impact of your training on trainings on being able to expand diversity in the life sciences?
James, do you want to? I was like, in
I feel like this
3-year
is the goal,
grants,
right?
yeah. I mean, well, and I think it depends on how you define diversity. It depends on what you define, right? Like a success. Is it moving people directly into those jobs? Is it retaining people in those jobs? I think is a really where we're a little bit more focused, because I think there's a lot of drive to, interview people and bring them in, and then they get brought into environments that are not actually going to support them, or they're like, nah, this doesn't work for me, and then they leave. But yeah.
So I guess in a way, we don't have a definitive answer on how much we're improving diversity, but we are hoping to improve inclusivity, and we are regularly assessing how people integrate into their organizations and find a space to learn and anyway make a space for themselves. And so that we're monitoring very closely, and we're assuming that will help all different sorts of populations create, anyway, find their way into the bioscience arena.
Yeah, intuitively, it seems to me that would work. I'm just interested in, how you measure it.
Yeah, I mean, I think a lot of what it has come down to so especially the integrated fellows program that we have, where we have these trained mentors with the city college students, is we're really looking at sort of what's the level of communication that's going on between them, what's the level of clarity, how comfortable are students in having difficult conversations with their mentors, right? Like, what is the trust level? And now, while this is not like a direct measurement of whether they're success and impact is such a really difficult thing to get to, but at least it's a piece of being like, okay, things are clearer. Like this, we put this activity in place, you tried it. Does that seem to you have more clarity? Do you trust your mentor? Do you feel that you can have a difficult conversation with your mentor? We have asked some questions around sense of belonging as well. But yeah, and still small cohorts too. So.
Thanks. Joe, why don't you ask your questions? I see some questions and conversation in chat, but maybe you could unmute yourself and ask your question.
Oh, thank you. Sorry. This is Jo Wu from Fullerton College. And I was curious because you had very small groups of students working, and it looks like you were able to cap your training to 12 students per lab class. And we have a really hard time trying to push that through CurricuNet. Most of our classes are supposed to be 24, and I just don't see how we could get enough support for each individual student that way.
Yeah, and that's a great comment, because we fight this battle, and then upper administration changes, and then a couple of years later we have to fight it all over again. so for our classes, typically for any of the technical classes that we're teaching here, and this is from introductory lab methods all the way through our advanced topics, it is pretty much the one instructor. And the biggest impact we had the 1 year we were teaching Biotech 1, there's a lot of really technical stuff that's going on in there, you know, from use of the equipment to access to the instructor. And we had been forced to let 18 to 19 students in that class. And the dean pretty much had a revolving door of students in there complaining. Not only could they not get to access the instructor when they got stuck and needed help, but they also couldn't quite get on all the equipment they needed to because there was 17 other students in their way. So that was the last time, and that was right before the pandemic, we won that as capping our classes at 12, because that's what one instructor can handle. That's what our equipment that we have can accommodate and still allow everyone access to it. I've been here at the college for almost 14 years now, and I think I've already fought that fight at least 3 times. I would expect, you know, a couple of years from now, I'll have to fight it again. So it is an ongoing thing. But if they don't provide any kind of like a TA that can be in the class with you, that's a lot of people to try and manage that you're trying to train in a very technical, demanding field that, you know, they don't get the answer to their one question. You might affect how successful they are 2 years from now because somebody couldn't correct one little thing because they couldn't get access to the instructor. So it is, Joe, I hear you. It is an ongoing thing that we battle with. And, you know, additionally from that, when we don't have a full class of 12, you know, we have rules on, you know, when they're going to cancel classes or make instructors teach for low enrollment pay. And they base those on, you know, double digits. Oh, you got to have double-digit students. Okay, well, I don't think that's quite right because it should be a percentage of the class, because if it's double digits for a 24-seat class that's only lecture, it should be a similar percentage, you know, 50% should be, and so that's something that's an ongoing battle here. And it's, but I hear you on all of that.
Oh, sorry.
Go ahead.
Do you ask the industry advisory board that question? Because we've been trying to put that onto our advisory board the last 2 years so that I can get the answer in the minutes because I'll ask them specifically. So how many people do you think should be trained on one tissue culture hood, right? And
Yeah.
trained.
Yeah, and I mean, that's
if you want
definitely
someone to go on
something.
record, just email us and we'll send you something
Okay.
useful, me and Carlos.
Thank you.
Yeah, that is definitely something on our advisory board. You know, class size is definitely a repeated discussion that we have, and that is retained as kind of more fodder for our fight when we need it. And I will say we do have incredible support from the industry here. And I think part of that is because we're embedded in an industry building, so we're not actually on a campus. So we're surrounded by industry every single day. And I think that that's incredibly valuable, not only for our students, but for the support that it can lend to, you know, limiting those course numbers of students in one class. Yeah, so.
Great,
Yeah.
thank you so much.
Actually, I'm wondering if people might want to enter the sizes of their classes in chat. Just as a quick poll, because it varies for us too.
Yeah.
Shoreline, it's been 12 to 16 classes I've taught. Austin, yeah.
Yeah. I will also just add, Joe, that the students, the pictures that you saw with students, those were actual high school interns. So that was not necessarily a college class that we were teaching. That was, we work with some of the local high schools and do internships a couple times a week for some of these students that just want to kind of test the waters to see if this is something they might want to explore as a career. And so we usually have a pretty small number of folks for that too, just because again, we have limited staff here on, you know, all the things that we try to do. We can't do everything. As much as we want to, we can't do everything.
Thank you. I'm really impressed.
That sounds like a great program. It's neat you're doing that. More
It's,
questions?
you know, it's fun working with the high schoolers though. I mean, they got a whole different perspective on playing around in the lab. It's great.
Oh, I could see that.
I had a quick question for Betsy and T. So I noticed and was intrigued by the fact that you guys had a TikTok channel that you were using that was related to the program. I'm just wondering, is that content being developed by your students? Are you somehow integrating something into your coursework that like prompts them to like, hey, you guys, let's create some stuff for, you know, for people who are outside of this program to learn about it? How's that work?
T, I'm gonna let you take this one.
So yeah, it depends on the population we work with because it's transient. So we started the TikTok channel last summer with the high school interns that we had, and they were in charge of it. They named it, they came up with a strategy, which would be like, how frequently do we post? What type of content do we post? And so they created a lot of that content. And then after the summer, in the fall, we talked about how there's a lot of users in the center who want to know just about different techniques or different spaces. And so the content kind of changes and shifts to a little bit of that, like basics of biotech. And then, and so it does change, but it is meant to complement whatever it is that the needs are for that learning population at that time. And we are hoping to do a lot more genome editing based TikTok to supplement the course that'll be released this summer.
Yeah, I mean, it just seems like a neat way of outreaching to, you know, to different populations. The question about like, have we increased diversity in our program? I feel like we could still work more on outreach. I believe that our classes absolutely do reflect the diverse population of our students that already exist at City College. And our focus, rather than like, can we get, you know, like 20% more Latinos into this class? Our focus is more like all of you feel like outsiders coming into this field, and we're like focused on like, have we been able to increase your sense of self-efficacy? Have we been able to increase your sense of belonging in science or your science identity? Do you have more agency in pursuing this career? And on those levels, I'm like, we most certainly have seen improvements on all of those metrics. And I hope that creates a kind of like launch pad for people coming from many different backgrounds. That's always been our focus, is like your background. How do you not assimilate, that would be a poor use of words, but find a space for yourself. And but still that extra step of like, yeah, this is working, but how do we find people who are not even considering biotech? And that still is like, we're hoping there's a bit of a trickle of like, well, you know, so-and-so has done well in this program and I know that person, maybe I could, you know, that's fine. But like how we outreach in general, again, to someone who's just never imagined it as a possibility. And I kind of like that TikTok approach. It seems like it would have some appeal.
I think you're going to make me download the app. It's one thing I've avoided.
You can see different videos just online too. You don't necessarily need the app.
Okay. Well, I might like to look at it anyway. There is a guy who has a Twitter account who's a DIY biologist who's been doing experiments with Ruby and making Ruby tobacco. I'll have to send you the link to that. Were there any other questions? I think it's probably about time for us to wrap up, but all right. Well, I'd like to thank our speakers, Karen and James and Betsy and T, and it's really wonderful. And we'll hope to see you again soon, and hope to see you in person at some point. Take care, everyone. And please fill out the evaluation.
Thank you.
Thank you, Sandy. Bye.